All right, so the first talk is by Peter Nico. So Peter is a scientist at the Lawrence Berkeley National Labs in Berkeley, California. So he's a staff scientist there. So his research focuses on understanding the mechanisms of metal redox chemistry and carbon cycling in soils and sediments. So the title of his talk is Unraveling Terrestrial Carbon Cycling at the Micron Scale Using Synchrotron Spectromicroscopy. Thank you, Peter. So you can start.
Great, thank you. So yeah, thank you very much for the organizers for having me and for the opportunity to present. And I enjoyed the talks this morning, and I'm looking forward to some more this afternoon. So as Satish said, I'm at Lawrence Berkeley National Laboratory. And before I go on beyond that, the work I'm going to present, as always, is a combination of lots of different collaborators and multiple funding sources from different labs and different light sources, but largely funded by the US Department of Energy. So as Satish said, we're going to talk about different techniques. The main technique I'm going to talk about today is STXM, scanning transmission X-ray microscopy. And Bhupesh provided some background on this, but just for those of you who weren't here this morning, I'll present again this little bit old, but basically accurate picture that the idea is STXM is a scanning technique where X-rays come in and they're focused down on through a zone plate onto a sample stage. Usually, not exclusively, but majorly, the sample operates in transmission mode, where the X-rays pass through the sample and the sample is scanned in an XY direction in order to image the sample, the entirety of the sample. It is used usually for the lighter elements, carbon, nitrogen, oxygen, that are very important in geochemistry, but also for the L and M images of heavy elements. Its claim to fame much of the time is that it's generally very high spatial resolution, tens of nanometers routinely, can be smaller than that. This is just a little bit of what the actual sample size sample system looks like. You put your sample on a small silicon nitride window and attach it to this aluminum plate, and the aluminum plate goes into the machine for scanning. So what you do once your sample's in the machine is that you take a transmission image of your sample, whatever that is. This is a soil particle here. Then you change the incoming energy of the X-rays, and you take another image of your sample, and then you change the incoming energy of the X-rays and you take another image of the sample, and so on until you have a stack of images at different energies of the same sample. But what that lets you do is that if you take any particular spot on that sample and look at the absorption of that spot over energy, what you get out is you get an X-ray absorption, the NextSAF spectrum of the element of interest or the feature of interest at that spot, this being a carbon NextSAF spectra. And the shape of this NextSAF spectra tells you something about the chemistry of that carbon. As Bhupesh pointed out this morning, there are difficulties. The sample handling and the thinness present challenges. The quantification is also difficult. However, that being said, we found it very useful for trying to understand what we call the micro ecosystem, largely in soils and sediments, thinking about carbon cycling. We know ecosystems can be organized and can think of organization at very different scales, from the continental scale all the way down to the organization of a given organism. The part that we find particularly interesting and challenging is what, as I said, we're calling the micro ecosystem, where kind of the microbial community, the chemistry that's controlling that community, and the physical structure are all roughly at the same scale. Or another way to think of it is this micro aggregate scale within inside soils, where all of these things are happening at once. And whether there's much still fundamental opportunity to understand exactly how these processes work. And what I'm going to show today is just a series of vignettes of different types of ways that we've used STXM to try to understand some of the carbon cycling at this micro ecosystem scale. So the first thing to really look at is, why bother doing all this small scale techniques when it's got all the hassles we talked about before? And the major point is that things are really different at the small scale. The understanding is different. I'll point out, here's a series of carbon NextF spectra of dissolved organic matter collected from a series of different rivers, some in North America, some in Scandinavia, some from peat bogs. And you can see that there are some differences in these NextF spectra by virtue of the different peaks being different sizes. And you can interpret and you can learn something from that. But I would also posit that fundamentally, they look pretty similar. On the other hand, and that's for a whole bunch of different rivers, when we took some dissolved organic matter from a river, Sacramento River in Northern California, and which is seen here, and precipitated it with iron oxides to bring it out of solution, and then looked at those iron organic matter precipitates with the Stixum, this is what we saw. So these are some of the ugliest particles in spectra that you've ever seen. But if you look at these 4 different spectra down here, these carbon NextF spectra, one thing you can say about them is they're very much not the same. So when you look at bulk chemistry, you're getting one thing, but as soon as you start to look into this micro scale in these complex natural systems and look at that spatial heterogeneity, you realize that the differences in carbon chemistry are much more dramatic and much more important than we see at this bulk scale. So there's real value in trying to get down to this micron scale and trying to understand what's going on and dealing with all the complexities you have to deal to do that. So another example I'm going to have is just looking at the changes at this scale over time. So we're interested in how microscopic redox fluctuations. So this is a rifle site in Colorado, which is famous in DOE lore for a lot of different reasons. But the water table at this site changes dramatically over the course of the year, going up several meters and down several meters over the course of the year, which means that you have much of the system that is cycling from oxic to anoxic conditions. And so we wanted to specifically replicate that and look at what happened. So we took a well in a reducing zone within the anoxic area of the rifle site, injected oxygenated groundwater into it in August, and then let it, so to create an oxygen zone within the anoxic area, and then let it return to its anoxic conditions to see what would happen over the subsequent months. Macroscopically, visually, this is what happens. In August, you have very bright red iron precipitates that then age and darken over time. We were interested in particularly what was going to happen to the carbon cycling part of this. So we can take our Stixum images, and on the top you see a series of Stixum carbon maps and the same location, Stixum iron maps in August 2012. And you can see these relatively nicely delineated carbon stalks that are all coated very uniformly with iron. We see in December 2012, it's relatively similar. The stalks are losing some of their integrity, but they're still generally coated with iron. As we look at, there's organic rich stalks, as I mentioned. As you look what happens as the months go on, in March 2013 and April 2013, we see that the stalks really start to fall apart. We see the appearance of what we technically term organic matter globules that are associated with iron 3 with those globules, and that the stalks themselves are starting to become carbonate rich and more associated with iron 2. So we're seeing the evolution of the carbon over time that was generated when they oxygenated the environment. We can look at, this is just TEM images of what some of these stalks look like again in August 2012, and then 9 months or so later, where they go from a very nice iron oxidizing bacteria coated with organic matter coated with this iron oxide to the decomposition, remineralization of both the carbon and the iron over time. If we look at the carbon NextF spectra that we can get from these individual stalks over time, not exactly the same stalk, but example stalks over time, we can look at how the peaks in this spectra change over time. And you can basically see the diminution of the more reduced area peaks. This peak is getting smaller, and the increase in the more oxidized peaks and the increase in the carbonate. So we can see over time that the carbon in the stalks themselves is becoming oxidized. Meanwhile, we can look at these other types of carbon that show up over time, and we can see that we develop actual iron 2 carbonate precipitates, where here's the the carbonate spectrum in the NEXAFs with almost no organic component. And we see that our organic matter globules, very fancily termed, as I said, associated with the iron 3, are still there, even in March and April. And they are the least oxidized and look most like the original carbon material. And the organic matter globules seem to be oxidizing more slowly or not at all compared to the rest of the carbon. So when we relate this to a kind of a conceptual model, we see that we're doing the oxidation. We're transferring electrons from the iron 2 to the oxygen and to the carbon dioxide in the system to generate organic matter and iron 3 material. When we return it back to reducing system, the electrons are basically being shuttled back from the organic carbon to the iron 3, reoxidizing that organic carbon. But some of the organic carbon remains reduced and associated with iron as iron organic matter precipitates, what we're calling our iron globules. This is important because we can relate this to other frequently used bulk modes of measuring carbon dynamics in soil, specifically this pyrophosphate extractable carbon, which is a net bulk selective extraction, but it's very commonly used. And we see at the same time period of our increase in our iron globules, we're seeing an increase in pyrophosphate extractable carbon from the system. This was our way to try to link what we were doing at the small scale to the large scale, because we know from other use that pyrophosphate extractable carbon on a C14 aging perspective tends to be significantly older than the total carbon in the system. So there's something functionally important about pyrophosphate extractable carbon, and we can relate that to what we saw in this oxic-anoxic cycle, that as you're turning this oxic-anoxic cycle of electrons going on and off and fixing carbon and reoxidizing carbon, you're not getting complete recycling, but you're spurring off a segment of co-precipitated iron organic carbon that seems to be more resistant to oxidation and potentially therefore is contributing to the pool of the longer stable carbon into soils and sediments. Another example we have looking at the same kind of organic matter cycling in soils is really trying to correlate elemental cycles with carbon also at this micron scale. To do this, one of the things we do is we also bring in, to complement the STXM, we bring in the complementary technique of what we call nanosims, which is a scanning mass spectroscopy technique where the sample ion beam is scanned across the sample, secondary ions are produced, those secondary ions are measured in a mass spectrometer. We can do our nanosims on exactly the same sample that we do our STXM on. STXM can tell us the changing in the chemistry. Nanosims can tell us the changing in the ionic composition, isotopic composition, which allows us to follow certain groups of molecules because we can add in carbon or nitrogen as isotopically labeled and then follow the chemical transformation of that. So we first did this in an example where we're adding 15N and 13C labeled chitin extracts to a Pacific Northwest forest soil and looking at the incubation. And when it came to extractions, after 3 weeks of incubation, 70% of the carbon and 90% of the nitrogen couldn't be accounted for in either the assimilated, mineralized, or soluble pool. It all went someplace else. So the question we got involved in asking is, where did it go? What happened to the remaining N, and where did it end up? So this is a SEM image at first of some of this microbial mat laid down on a silicon nitride window for STXM analysis. The wonderful and terrifying part of working at this scale is there's all this complexity we're looking at right now, and we're going to this one little box right here and trying to understand what's happening with this chemistry here. And we can see this is a single transmission image. Here's a grouped image showing different types of carbon chemistry based on their spectra in this area. And we can see that those different colors give us different types of carbon spectra. So there are different types of carbon. We assign some of these spectra to those of the fresh hyphae based on their morphology. Those of the slightly decomposed hyphae, again, based on those morphology. And then the other two we assign through looking at standards. We look at a bacterial biomass standard and say that this group looks quite a bit like, not perfectly, but quite a bit like a bacterial biomass. And this group over here looks quite a bit like our excreted protein standard. So we're able to roughly assign buckets of carbon to these types of material we're seeing in this sample. We can then take we can show that we can do this multiple times, not a lot of times, because beam time is always limited and it's a difficult experiment, but we can do it enough times to show that it's not a single one-off experiment. We can then take those samples and scan them in the nanosims to look for the isotopic composition of those different types of materials. And we can see that the 15N composition of our different types of organic matter seems to change systematically with the type of organic matter from the fresh hyphae to the type of material associated with the iron mineral surfaces. We can only do this so many times with the STXM, but with that new hypothesis, we can then, in the nanosims, scan all of the samples with iron and compare them to all of the samples without iron and get a statistical representation of the N15 composition, which does bear out the hypothesis that the N15 composition on the iron is higher than in other places. So now we've traced the iron, and we've traced the nitrogen and we've traced the carbon, and we can build a conceptual model that we're having these living hyphae with certain N15 composition, turning into decomposing hyphae, bacterial protein type material, and ultimately stuff deposited on the iron surfaces with an increasing concentration and retention of the N15 label during that carbon transformation. So, and then the other thing I would just want to point out quickly is I've talked in a couple of different STXM examples here, but I also wanted to point out that there is another synchrotron-based technique that is good for looking at carbon at the small scale. And that's Synchrotron FTIR, which is Fourier transform infrared spectroscopy, that really does the same thing that your lab infrared spectrometer does, but it does it with a much higher signal to noise ratio at a much smaller spot. So in this case, we're trying to understand how manganese is important in the decomposition of litter of leaves and needles deposited on a forest floor. There's bulk scale information on that manganese tends to increase the rate at which mass is removed as needles decompose. More manganese, the faster they decompose, less manganese, the slower they decompose. But there wasn't really much by way of mechanistic information at the small scale. So what we're able to do is take some decomposing pine needles in the Pacific Northwest forest that have been colonized by fungi, and looking at, here's a light microscope image of a thin section looking at the interface between the colonization of the fungi and the needle being colonized. Look at X-ray microprobe, hard X-ray microprobe distribution of the manganese, and noticing that if here's the interface, all the manganese is concentrated right around this interface location between the fungus and the needle being decomposed. And then use the Synchrotron FTIR images of those same locations to look at how the chemistry of the material is changing right at that interface, and notice not only the changes in the chemistry on either sides of it, but particularly at that interface, an increase in the carbonyl oxidation, so that we're getting right where the manganese is, right where the carbon's changing, we're getting this interfacial information. So we can take this idea that we have manganese scaling manganese cycling at the large scale, which is very important for litter decomposition, and take it down to this idea that at the small scale, we can look right at where there's a fungal hyphae that has manganese 2 and manganese peroxidase enzymes that's oxidizing the manganese 2 to manganese 3 and simultaneously oxidizing the organic carbon in that litter sample right at that very specific interface. And so those are, I think I'm coming right up on my 20 minutes. Those are my 3 examples of how we've used this small scale spectrum microscopic capabilities to try to build our conceptual model of what's happening here. I'll put 2 things out here at the end, which I think are interesting and could be discussed. So at great effort by Marco Kaluait and Jeremy Micron as a postdoc, made a cryo thin section of a soil microaggregate at this location. And then this was after incubating it with 13C CO2. So again, we can get a nanosims spectra of this area, and we can see certain hotspots, presumably active microorganisms that show up in this area. We can get a STIXM image of this area and look at the carbon in that image. We can see that where the microbes aren't really the major sources of carbons. They're huge other sources of carbon, but physically separated from those organisms. So it leads to interesting questions about how, when, and if these organisms can access this carbon, which is literally only microns away, but may or may not be in any way accessible depending on the structure of that microaggregate. And lastly, I'll talk about for discussion things that we are challenged by, which is the idea that scaling is hard, but potentially tractable. So we're dealing down here at this micron scale, and we're able to take a few images if we have a significant amount of beam time. And we're wanting to scale up to at least, let's say, a meter scale plot or a lab scale experiment. And we're trying to cover 5 orders of magnitude with this. This is a challenge, and I don't mean to minimize it, but I will also point out that there are plenty of experiments that take a few meter scale plots and then scale up to the size of the Sierra Nevada forests in California. And this is a 6 order of magnitude jump from the same type of scaling techniques. So there's things we can learn from large scale experiments to try to understand how we can do our scaling. I will say, to the point of this workshop, that it's very important to be able to do enough analyses to get something like a statistical representation of what you're looking at that small scale, and to have access to multiple different techniques in order to look at these things from different ways, as multiple people have pointed out earlier today. And so with that, I will finish, and I think we have a couple minutes for asking questions. Thank you very much.
Thank you, Peter, for a beautiful talk. Any comments, questions? Well, I have a question. So using STIXM, I know you can identify different pools of carbon, and I'm wondering whether we can improve the detection method somehow with a combination of techniques.
You mean just be, yes, I think we really should be able to. And as we talked about, sometimes the STIXM analysis is, the next steps analysis is ambiguous. Being able to look at exactly the same sample with something else, the IR or the nano IR would be very helpful. And I think it's often important to be able to do exactly what you just said, Satish. So I'm just agreeing that it is important to be able to look at it in different techniques. And I also would say it's important to be able to look at it with different elements, because sometimes you can look at the carbon versus the nitrogen versus the oxygen, or things like phosphorus or other elements to help you understand at that same spot the different elemental cycles that are going on to help you interpret all of this. There isn't a single way to get out exactly what you're looking for.
Right. Thank you, Peter. So maybe we'll move to Ethan's talk.